Hi everyone, welcome to CAPSA. Today's topic is about Pale Catechu. So Pale Catechu, another names or synonyms are Gambier and Catechu, and it is actually obtained from the dried aqueous extracts of leaves and twigs of Uncaria Gambir, which belongs to the family Rubiaceae. And now, what are the geographical sources? It actually present in Malaya, Indonesia, Borneo, Singapore, etc. Among these, Malaya is a indigenous place and cultivated in Indonesia, Borneo and Singapore, etc. Now, how they are collected and prepared? So, it actually grows as a climbing shrub. Okay, from the seeds they are propagated. The leafy twigs up to 50 centimeter in length are cut at an interval of 4 to 6 months from the 2 to 10 years old plant. Okay. After the collection, the leaves and twigs are boiled with water for 3 hours in a large pan. After removing the leaves and twigs, extract is concentrated to form a yellowish greeny pasty mass. Okay. And these semi-solid mass are cut into pieces like cubes and packed for export. Okay. Now we will see what are the chemical constituents present. So the main chemical constituents are Catechutannic acid, Catechin. Others are Quercetin, Pyrogalol, Gambir fluorescence, fixed oils, waxes, etc. Okay. Among these, Catechutannic acid is a kind of Fluorescence. But Catechin is a pseudotannin. And Catechin forms white needle-like crystals which dissolves in alcohol and hot water. But Catechutannic acid gives green color with ferric chloride. Okay. Now, what are the chemical tests? Okay. So, it mainly shows two type of chemical test or two type of chemical tests are performed. First one is Gambier fluorescence test. Second one is Vanillin hydrochloric acid test. Now we will understand in details. In Gambier fluorescence test, actually the chemical constituent Gambier fluorescence, which is present in the Pale Catechu, gives the fluorescence. But Black Catechu does not give this kind of fluorescence. It means Black Catechu so the negative test. Okay, and this type of test is due to Gambier fluorescent kind of chemical constituent which is present in the Pale Catechu. Vanillin hydrochloric acid test. In this test, due to presence of catechin moiety, the drug shows pink or red color with a mixture of vanillin, alcohol and hydrochloric acid. Okay, and the composition is vanillin, alcohol and dilute HCl. This composition is in a ratio of 1:10:10, gives the pink or red color. Okay, due to the presence of catechin. In this, fluorescence are shows due to the presence of Gambier fluorescent moiety. Okay, fluorescent moiety. Clear? Now, what are the uses? It actually used as local astringent. In diarrhea, it is used for chewing with betel leaves. It is used in dyeing and tanning industries. Also, it is used. Now, what is the allied drugs? Okay, so allied drugs are known as Kach, synonym or Black Catechu, which is obtained from the heartwood of extract of heartwood of Acacia Catechu, which belongs to the family Leguminosae, and it occurs in black color or porous mass and taste is similar to Gambier. And this Kach or Black Catechu contains catechin near about 2 to 12% and fluorescence which is 25 to 33%, and others are quercetin, moisture, etc. And this catechin or Catechin is not identical with the Gambier. Okay. Now we will see the chemical structure. So this is the chemical structure of catechin. In this, 1 and 2 benzene rings are present, which is attached with a heterocyclic moiety, that is dihydropyran. Okay, that is a heterocyclic moiety type C. Okay, this is the Uncaria Gambir plant. Okay, and leaves and twigs part are collected from this shrub. Okay, this is Pale Catechu cubes. After the preparation, it looks like this. Okay, so this is a kind of herbal drug named Pale Catechu, which is very much important for pharmacognosy. If you like my video, please do like, comment, share and subscribe. Thank you.